So one of the most fascinating discoveries in the modern genetics is that we as humans share around 99.9% similarity. But despite of this genetic similarity, we have populations from different regions of the world, and we all exhibit different patterns in genetic variations. Some are white, some are black, some have long hair, some have short hair, and even the way we fight diseases is very different. So one question will arise: how such a small difference in genetics bring about those huge differences in us? So in this project, we are going to answer that question through a reproducible bioinformatics workflow, and we are going to use publicly available genomic data from 1000 Genomes Project. So before we begin, I might share some few fascinating facts about the human genetics. So did you know that currently, as of 2026, there are more than 8.3 billion people that are living on the earth, with India and China covering up to 36% of the total human population. So for many centuries, many researchers relied on physical characteristics such as one's skin color or facial features or language or geographical location to just study the human population and study how diseases actually spread, and trying to find some keywords to specific diseases such as sickle cell anemia, which is most common in Africa and other sub-African regions. So later in 1953, when James Watson and Francis Crick discovered the double helix structure of DNA and also revealed that we store genetic information and it can be passed from one generation to another. So later on, around 2008, this is when the international collaboration began at a large scale to study one of the most asked questions, that where humans have begun. So this is when 1000 Genomes Project was launched, and it became one of the most comprehensive public catalog of human genetic variation, where different genetic information from around the globe was brought together and analyzed from thousands and thousands of individuals. So this dataset is publicly available. So I'm just going to do an investigation on some of the principal differences that we carry as humans, and try also to figure out some ancestral attributes that we might have using this real genomic data, which is publicly available. And as well, I'm going to document everything in GitHub, and I'm going to put the link in the description. So if you want a reproducible set of the project, you will find it in GitHub. So let's begin. So the first step, as usual, is data acquisition. This is where we are going to obtain the genomic data for our analysis. Okay, so we go to your favorite browser, we search for 1000 Genomics, and then we go to the website. Okay, so this is the website for 1000 Genomics, where you can find various datasets. So I'm going to click the search button and find the latest dataset that was recently published. That is 2013-0502. Okay, I got 3,859 matching data files. And actually the dataset that I needed in this case was the chromosome number 22 dataset and the metadata file in the VCF file, that is the variant core format. But then see what happened. I got 3,859 matching files, and it was going to be a very much difficult hurdle to search through this data to find the specific data that I needed. So I ended up using the command prompt to download the data. It's one of the most effective ways of downloading this such kind of data sets. Okay, so I'll go and hit the command prompt, and I've already looked for the correct URLs. So I'm just going to paste. So it ended up taking more time because the files are pretty huge, because we have millions and millions of genes from different individuals. So the next step is variant exploration. This is where we are now going to understand the genetic variants in our data. Okay, so as usual, I don't enjoy explaining how I write my R scripts. So I pre-write my scripts to make the workflow a little bit easier. Okay, so the process is going on, and it is reading about 1,055,000 characters. But unfortunately, I have done a very big mistake in this project, because suddenly my machine froze and it ceased working. and I tried to figure out what was wrong. So I ended up using another machine to correct my mistake because the other machine froze during the process. So I ended up doing some further data trimming where I've significantly reduced the dataset, and I've filtered some of the information that might pollute our data as well. So I urge you one thing, before you try to analyze these types of datasets, I urge you to first verify if your machine can handle this dataset. If you are a laptop user, I recommend you something with more number of cores, because this is what the analysis actually use. So I had to return to the old machine with the new datasets that were trimmed. So now let's run them, and this is going to be a success. And this one as well took almost 15 minutes. Okay, so we are done. So the very last chapter, which is quite my favorite, is the data interpretation. This is where we are now going to see what output we have got from this dataset. So let's begin the data analysis. Okay, so since I started making these videos, I have discovered one thing, that bioinformatics data is not that much easy to explain what you have found, because it contains millions and millions of information. But I will try my best to explain what we have found. Okay, so let's start with the MAF distribution. So the MAF simply means it's a minor allele frequency distribution. For you to easily understand how the MAF plot works, suppose we are analyzing a group of 10 members and you are looking at their DNA position. So suppose we are just focusing on the A and the G nucleotides, and the sequence contains around 7 or 8 A nucleotides and only 2 G nucleotides. So we are going to say that A is more common than G. The A nucleotides are going to be called the major allele, and then the G nucleotides will be the minor allele. So when the MAF plot distribution is closer to 0, it means that there is a very rare variant, which means the data is densely spaced, because if it was almost similar, we should have found that the ratio between the alleles should be almost close. So looking at our data, we have seen that there's a huge concentration of variants which are very much close to 0. So what we have done, we have applied a 5% threshold cutoff, which is shown here by this straight line, and it retained variants which are above that threshold, and this has reduced our dataset from 99,000 variants that we have to around just 9,000. So overall, the MAF analysis simply showed us that most of our genetic variants in our original datasets were very rare. And when we say the genetic variant is rare, we are just simply trying to say that there are very few people in the whole dataset which have a particular version of DNA that is almost similar alleles. Okay, so due to the computational restriction that I have suffered before, I was not able to actually have a full analysis of all the datasets. So the next result is the MDS population structure, which simply means multidimensional scaling distribution. So since we have reduced our data to around 520 people and only 9,000 genetic variants, if we had tried to compare every person's genetic profile directly, it was definitely going to be impossible with the current computational resources that I have. We are just going to calculate the genetic distance between the individuals that we have analyzed. So what the MDS distribution does is it tries to map the data into a 2D structure to show us which people are genetically similar when placed together. So in this dataset, there's a clear separation between the groups of individuals, which means that the genetic variations we were analyzing contains a detectable population structure. So at this point, we can conclude that among the 520 individuals which we have represented by thousands of genetic variants, we have successfully been able to separate them into different clusters based on their genetic variants. Okay, so the last most important result that we are going to analyze is called the dendrogram. So the dendrogram is basically a tree that shows which individuals have similar genetic profiles. So the dendrogram was generated by calculating the genetic differences between each individual. Those individuals that have more genetic similarity were joined together first, and then those smaller groups were gradually joined into larger groups. So basically the dendrogram is just showing us the genetic variance as well among the people, and since we have constituted our dataset up to only 520 people, the variance is now a little bit limited. If you have enough computational resources to do the whole analysis pipeline, you can safely run the whole scripts and get more detailed results from this analysis. Okay, so in conclusion, we were hoping to analyze a more detailed number of individuals which were representing multiple human population, but due to computational constraint, we've only managed to examine genetic variation which have been presented by only 520 individuals. So however, in our analysis, it does not determine the ancestry of an unknown individual. Instead, it has demonstrated how we can use computational methods to actually identify and visualize genetic relationships between known populations just by using their genetic variances. So rather than asking, can DNA tell us where we came from, our analysis has significantly shifted to show us something more precise, that DNA contains patterns of variation that can allow us to group people into different clusters and to investigate how the human population can be genetically structured and related. So thank you if you have reached to the end of this video. See you in the next one.